So, hi. So, first of all, tell us exactly who you are, because
I'm
I,
I'm
I read
I'm
an interview some months ago, but it wasn't you. I think it was your wife, Jill.
could be could
Yes.
be.
So, who
Um,
you are and what you did in the last 80s.
okay, right, the RNA story. So I'm Robert Malone. I'm a physician scientist, and I live on a Lusitano horse farm. We breed Lusitanos that we have brought over here to Virginia from Portugal. And I'm very glad to talk to you. Thank you for the chance. And what did I do? I've done many things in my career, but when I was very young, 28, 29, I was a graduate student at a place called the Salk Institute, which was created by Jonas Salk, the polio vaccine expert, which is located in La Jolla, California, on the cliffs overlooking the Pacific Ocean. So, which I grew up in California, which is why I love Portugal, is Portugal faces the ocean in the right direction. And it has mountains at your back, which is the way things should be. And you can see the sunset over the ocean. So I was at the Salk Institute in La Jolla and working in a very competitive, incredibly competitive postdoctoral research lab as the only graduate student. And the invention that matters for this talk today is that the whole suite of technologies that underpins the modern mRNA vaccines came off of my bench and I guess out of my head during the period from 1987 to 1989. First at the Salk Institute, and then at a little startup company that was kind of a little bit across the street called Vical. And those discoveries led to a manuscript in the Proceedings of the National Academy of Sciences, which laid the groundwork on RNA, mRNA for delivery into cells, the technology to use for that, which is to coat it with fats or lipids, and how the structural elements of the RNA necessary to make this work, how they interact with each other, and the use of the manufacturing process to make large quantities of it, which had to be developed, and the use of this for delivering RNA into cells and tissues in animals, both for RNA as a drug and RNA as a vaccine. And it's a fascinating story of different people, and it's a series of discoveries that didn't necessarily have to happen. The expression "chance favors the prepared mind," and I was very much surrounded by amazing people and scientists at the Salk that helped enable all this. So it was being an open mind in the right place at the right time and having a series of findings. Interestingly, the other genetic vaccine that technology that is being used right now, recombinant adenoviruses, really came out of the same laboratory. So the senior postdoc in the lab when I was there was a gentleman named Dinko Valerio. Dinko created a company called Crucell. Crucell was sold to Johnson and Johnson, and that's the basis for the J&J vaccine. And there are people who object to me calling these both the RNA and the adenovectored vaccines gene therapy applied to vaccinology, but that's what they are. And they both came out of a gene therapy lab, and they both were the consequence of an experiment that was done that showed that gene therapy was having problems with immune responses. And I don't know if you have this expression in Portugal, but we certainly have it in California. when life gives you lemons, you make lemonade.
Yes, yes, we have this.
So the starting of all of this was really because of the finding that gene therapy was going to have problems with immune response. And I was a young, partially trained physician in this laboratory, gene therapy laboratory, and I realized that we could make lemonade. We could use these gene therapy technologies to make vaccines. So that was the start of it all. Thanks for asking.
So for an ordinary person, what is the difference between a regular vaccine and these ones? What is the difference? Because
Great
it isn't
question.
the same thing, and the people think it's the same thing. Actually, the purpose is the same, but the way to there is different. That is right.
It is precisely right. And thank you for asking. And I have had many interviews lately, and no one has asked that question. So thank you. Regular vaccines, whatever that is. So the idea of a vaccine is that, and there are kind of two categories. There's vaccines that we use for therapy, like some people talk about cancer vaccines, okay, that will boost your immune system's ability to attack a cancer cell.
Okay? But I think I'm sorry, I think the ordinary people think like a measles vaccine. It's the same.
I'm getting there. I'm getting there. Okay. So the first big division in the vaccine hierarchy of things is vaccines that are therapies that get your immune system to attack something after you already have the disease. And then there's like measles vaccines that are vaccines to protect you from something you don't have yet, or protect your children. Measles, mumps, rubella, polio, smallpox, these kinds of things, right? Influenza. So let's take, do you use influenza vaccine in Portugal?
Yes, every year, yes.
So let's think about influenza vaccine just as a way to think about ordinary vaccines. Influenza vaccine is made basically either from a killed virus, influenza virus that's grown in eggs, or you kill the influenza virus, you isolate it, you kill it, and you break it apart. And you mix it with something that helps make your immune system work better against that pieces of virus. So an ordinary vaccine is pieces of virus or pieces of a bacteria that's mixed together with something that stimulates your immune system and gets injected. And so these are collections of virus pieces that are in a needle that you put into your arm or your baby's arm, or maybe they might take it in their mouth. It produces, there's a very specific amount of protein that's in that mixture. So we know when we put it into your arm exactly what's in that syringe, how much protein, we call it antigen. There's a bunch of fancy words that go with this, but what it comes down to is there are fragments of the thing that you're trying to protect against that you mix with something that stimulates your immune system, and it's injected typically into your muscle. And your immune system recognizes this and makes a response as if you have been infected, but you haven't, because the parts that were injected to you can't make a whole virus. They're not alive. They can't replicate. So that's the usual vaccine, right? Do we agree?
Yes.
So with these gene therapy based vaccines, recombinant adenovirus vectored is one. So that's Sanofi, Oxford, J&J. Those are all adenovirus vectored vaccines. Let's talk about those first. What those are is a cold virus, a live cold virus that we have used modern technology and taken out a little piece of the DNA. In this case, those are DNA viruses. We've taken out a little piece of the DNA, and we've taken a piece of DNA that would produce one of these proteins that might be in, say, SARS-CoV-2. So in this case, we're going to talk about the SARS vaccines in these examples. We take the sequence that would make a protein that's on the surface of the SARS-CoV-2 protein. We call it spike. It happens to be the protein that the SARS-CoV-2 virus uses to attach to a cell. Okay, so it has this little thing sticking out. If my hand is the virus, it has a little cluster of proteins that are spike, and it comes up to a cell and it finds a receptor that's just perfectly matched to fit those three proteins. And then they undergo a change in structure and they are able to poke in and enter the cell. Okay, so that's spike. And so what was done is to take the gene for spike and put it into the cold virus, the adenovirus, in place of, got rid of some of the parts of the adenovirus that we didn't need, put it in there, and then that cold virus is made in a fermentation tank, like you might make beer, same kind of technology, but it's not yeast, but it's the same idea. And then that's purified, and that's put into your arm. And that cold virus is able to infect your cells and put its genes into some of your cells and make those cells become little factories that make spike protein. Now, this is what a regular virus, what it does is exactly that. The SARS-CoV-2, you breathe it in or you get it in your mouth or whatever, and it infects you, and it goes and it attaches to your cell, infects your cell, and it takes over your cell and makes that cell start making viruses. Okay, the adenovirus does kind of the same thing, but it doesn't produce more adenovirus. It only makes your cell become a little factory for making spike protein. And that spike protein then stimulates your immune system, and it stimulates it in ways that are in some ways better than if you just injected the free protein into your arm, because from the point of view of your immune system, those cells look like they've been infected. And so because they have spike protein on their surface. And so as far as your immune system goes, this is the real thing. And it makes two kinds of immunity against that. One is antibodies that we all understand about, antibodies. We've heard about antibodies. And the other is something else that you may or may not have heard of, cytotoxic T lymphocytes. So these are killer cells. And it's really good to have those if you're having a virus infection, because they can search all over your body and find little places where there are infected cells that are infected with SARS-CoV-2, and they kill those cells. Remember I said that the virus turns your cells into little manufacturing plants. So instead of turning out dinnerware, they're turning out viruses, and we don't want that anymore. So your body's immune system makes these killer cells that go hunt them down and kill those cells. So that's good. Two kinds of immunity, antibodies and cellular. Okay, now let's talk about RNA vaccines or classical DNA vaccines, but RNA vaccines is the topic here.
There are the Pfizer and Moderna, right? This one.
Correct. And CureVac is trying to bring up another one. Okay. And I'm sure that over the next few years, there'll be many others. And maybe someday there will be a RNA vaccine company in Portugal. I hope that's the case. Then I could move to Portugal and help with that company. And I would be happy, and we could have our horses in Portugal. So that's what I'd like to do. Let's do it. RNA vaccines. In the case of RNA vaccines, what you do, I'm gonna teach you something. Forgive me, I'm a professor. I used to be a professor and I can't help but teach. So in biology, we have this thing called the central dogma, and you have to kind of understand this a little bit. The central dogma of biology is that DNA makes RNA, RNA makes protein. So in the case of spike, if it's an adenovirus, the adenovirus puts DNA into the cell, the DNA makes RNA, the RNA makes the spike protein goes on the surface of the cell, and the immune system does its business. In the case of an RNA vaccine, you make the RNA in a test tube, in a industrial manufacturing plant, like you might make a chemical or a pharmaceutical, like you might make aspirin, okay? So with all of the controls, I mean, Portugal has their own pharmaceutical industry, and you do make drugs there. And certainly you know about the pharmacist. When I go to Portugal, I love to go to the pharmacist because I can get things over the counter. You understand about drugs, and the RNA is just another drug, in a sense. It's made in a chemical synthesis process. So it's not made biologically, it's made chemically, and it's purified. And then there's kind of some special magic that was invented by a group at the University of British Columbia, the latest technology that's made all this happen. It's the group of Dr. Peter Cullis. And they have had some, I mentioned that back in the day, in the late 80s, I was working with fats that would coat the RNA. And they used a certain kind of fat, a cationic lipid. And what Peter Cullis and his group at University of British Columbia did is they advanced that technology. And they advanced it so far that this really works well in humans now. This ability to coat the RNA, wrap it in a fat, and make it so that it can, if you inject it, it will go into the cells of you or me. I took Moderna, that's a separate topic, but it worked for me. It'll work for you. It works for all of us, thanks to Peter Cullis and his team in British Columbia. So they learned how to better wrap it with these fats. So this is all happening in big chemical manufacturing processes, lots of stainless steel and pipes and tubes and pumps and things like that. And it produces a mixture of very, very tiny particles. They're so tiny you can't see them under a microscope. You have to use special equipment or an electron microscope. You can see them, but they're very, very tiny. They're a few nanometers. So that's, we're all in the metric system in Portugal. So micrometers, nanometers are 1,000 times smaller, very small stuff. So this mixture of really tiny particles with RNA at their core, wrapped in lipids, is injected, and they have been engineered in such a way that they're able to slip directly into the cytoplasm of your cell. So for those who aren't cell biologists, forgive me, a cell is kind of like an egg. When you fry an egg, there's the yellow part, and if that's a cell, remember, cell's a teeny tiny thing, but the yellow part of the egg, when you go to fry it, it can be thought of as like the nucleus, and the white part of the egg is like the cytoplasm, and the very edge, when you're frying it, that gets all slightly brown and crinkly, you could call that the cell membrane. Okay, so an egg as a metaphor for a cell, and the thing is that barrier that makes the yellow part of the egg stay put is if we're imagining a cell like an egg, that barrier on the yellow part of the egg is another membrane barrier that keeps things from going in and out. So if you put, say, DNA into a cell, it's got to get through the first barrier, which is that outside part of the egg that's a little bit brown and crinkly when you fry it. And then it's got to get all the way through the white part. and then it's got to get to the yellow part, and then it's got to get across those barriers and inside, and then it's got to be made into RNA, and then that RNA's got to get back out into the white part, where the protein translation machinery is, the stuff that takes that message and turns it into protein before it can ever do its business. And so the lovely thing about there's two fantastic things about the RNA as a drug or RNA as a vaccine idea. The RNA only has to get into the white part of the egg. It doesn't have to go all the way to the yellow part. So it's a lot more efficient. The other thing that's really good about it, it's a safety thing, is that RNA generally doesn't stick around too long, maybe a few hours. And so it gets into the cell, it makes the protein, and then it gets broken up. And it's no longer making protein. So it's more like a drug. A drug you take, and if you have an adverse event, if you feel bad because of the drug, you stop taking the drug. Similar kind of thing with RNA in a sense. Unlike DNA or gene vectored, adenoviral vectored stuff that make the protein for a very long period of time. The RNA gets in there, it does its business, it makes the protein, and it's gone. So that all sounds really good. And it turns out that it works remarkably well for vaccination purposes. And it looks like the technology that Peter Cullis and his colleagues have advanced is so powerful that it's going to open up a whole bunch of new applications that are basically, we now have a new kind of biologic material, RNA, that we can use for drug development. So now you asked me to do a complicated thing, explain the difference between kind of old school vaccines and these gene therapy based vaccines. And you could ask, why are they gene therapy? Well, RNA, some people might say, well, RNA is not a gene. Genes are all DNA. That's not actually true. RNA is genetic material. And if you're a RNA virus, for example, say SARS-CoV-2, all of your genes are in RNA. They're not in DNA. Okay, so yes, RNA can be, you can have genes that are on RNA, you can have genes that are on DNA. And we're using this genetic material that's coming from outside, you're synthesizing it in chemicals, and you're putting it into the cells of your body for therapeutic purposes, in this case, prophylaxis, prevention, but it can also be because you want to express a protein that's going to be helpful. So that's RNA as a drug. And this is the thing that's got a lot of, I got to tell you, I'm amazed at how much money is in play right now, investment money, now that this technology has come to this stage and it's been administered all over the world. There are billions and billions and billions of dollars that are being invested in this tech to build the next generation of RNA drugs and RNA vaccines. So it's a very exciting time for me. I never thought I would see it. I thought I would be long gone and in the ground when this happened, but it's happened. It's a vision that I had over 30 years ago, and it had all kinds of obstacles, but here we are, and now it seems to be working quite well.
Okay, so I want to ask you, because there is this study provided to Japanese authorities by Pfizer, says that the capsule, lipidic capsule of does not stay in the shoulder, and was found in the many places. I want that you explain what that means, what could do long term, because they say, there is all over the place. Okay, but what it could happen? Because they are talking a lot about fertility, female and male. Can you explain that, please?
Yes, and I apologize that I can't speak Portuguese. Maybe someday I will be able to.
My English is terrible, so don't worry.
No, your English is fine, and it's much better than my Portuguese. Plus, if I could understand Portuguese, I could understand fado, so that would be good. So I sometimes I talk too fast, so
That's
I'll
okay.
try. Okay, all right. So she's asking about. I'm gonna use some words that are a little complicated. They're regulatory words in regulatory language, but she's asking a complicated question about some key technical documents. And so I can't not use those words or we'll never get through this.
Simple words.
So what she's talking about is a Freedom of Information Act action that was taken by some Canadian scientists and physicians that were concerned about some of the adverse events that are observed. Adverse events is what we talk in clinical development to mean toxicity, bad things that happen. You know, soreness and redness is an adverse event, for example. Vertigo, your head spinning, is one kind of adverse event that happens with these vaccines sometimes. There are many others, and I'm sure we'll talk about those at some point. So some Canadian physicians that were concerned about the adverse events that they were observing in their clinics petitioned through a Freedom of Information Act to obtain what's called the Common Technical Document, or in the United States, we call it the Initial New Drug Application, which is the document which summarizes everything that was known by Pfizer about their vaccine drug product at the time when that document was submitted to the Japanese regulatory authorities. And there's a similar document submitted, I presume, but I haven't seen it, to the European Medicines Agency, which I think Portugal, I don't know if Portugal maintains their own regulatory authority or they just rely on EMA. Do you know the answer?
I think that they follow what EMA suggests.
Yeah, so, yep. Okay,
but
so
we have the information, but I don't know.
Yeah, so the Japanese had received this common technical document from Pfizer many months ago, as had the EMA. So this Japanese version was released. Normally, this document is never able to be seen except by the regulators and the company. But for some reason, Japan decided to release it. And in that, I received that document within a day of when it was released. I was asked to review it for a US online publishing site called Trial Site News. And I also was asked to review the letter from the European Medicines Agency, which is the summary letter of their review of a similar document. So I was given these two things and asked to review them. The Japanese document has some tables in English. Everything else is Japanese characters, and I can't read Japanese, so I can't read that. But I can fortunately read English. And one of the English tables has a summary of the biodistribution experiments. So what is biodistribution? These are the experiments that a pharmaceutical company has to have for any drug, where they administer it to animal models before it's ever used in humans, typically rodents, rats, or mice. And the drug is administered to the animal, and you track where it goes in the body of the animal and how things are concentrated or not concentrated in the body of the animal, because it teaches you about what might be problems in humans. This is part of a larger group of data that is required for any new drug or vaccine in order to be approved to be used in humans. One of the things that was, so as I reviewed this, I found some things that I was not comfortable with. was a little alarming to me in this package. And I checked with another regulatory, a more senior person, and he also concurred and found some other things I'd missed. So by comparing the Japanese document and the letter from the European Medicines Agency, what we found was that Pfizer at that time had not completed the standard reproductive toxicology studies, had not done genotoxicity. This is whether or not the drug will affect DNA or RNA and cause problems, or might cause problems. The biodistribution studies were done non-GLP. What this means is that they weren't done to the quality standards that is required for a drug before it's administered to humans. And furthermore, the biodistribution studies were not done using the actual drug. They were done using an RNA that codes for the protein that makes the firefly tail glow. Now, I don't remember if Portugal has fireflies. Do you know fireflies? They're a little insect that flies, and they glow, and they use flashes.
Pirilamps,
I think. Good, thank you.
Maybe.
Okay, so they're up in the trees in the summertime. Glowing.
Okay. Yes. Right. Pirilamps. Yes.
Okay, so this is the protein that they use to make that flash of light.
Oh.
Okay, and it's called luciferase. It has nothing to do with the devil. It's just what they called it when they cloned it. And it's a really good way to see whether or not you were able to introduce a gene into a cell or a tissue and have it made into protein. Because if the luciferase protein is made, then you can add the substrate, which is called luciferin. Luciferase cleaves luciferin in the presence of ATP, and it releases a photon of light.
Okay.
And we have photomultiplier tubes that are really good at detecting photons. Yay. And this results in a very linear assay where the amount of light produced is very much a function of how much protein there is. And so you can do the reaction, include controls, and you'll be able to say, there's this many photons, and that means there's that much protein that's been made in that tissue. Okay, cool assay based on fireflies. So this is what Pfizer used. Normally, you would be required to do your tests for delivery and distribution and gene expression. You would be required to do it using the drug. But they didn't do that. So they didn't express spike. They used luciferase as a surrogate. And I mentioned that normally you would take, let's say, a mouse, and you would inject your mixture encoding luciferase, and it would go throughout the mouse and it would make the protein. And then you would sacrifice the mouse and you would dissect out the tissue of, kidneys, liver, spleen, brain, heart, all those things. And you would put them each in test tubes and grind them up and test for how much light is produced, and you would know how much protein was produced. Now, there's another neat thing you can do with luciferase is you can take the mouse or the rat and directly inject the luciferin into it together with some ATP, and the mouse or rat will start glowing. And the photons will come through the skin, and you can take your photomultiplier tube and you can image the mouse and you can figure out exactly where the luciferase protein is. But it is the least sensitive way to do this. So you can imagine if you did that experiment, you would see where the highest concentration of the gene went, the RNA, but you wouldn't detect small amounts that went to the foot or the nose or whatever. Pfizer elected to use the least sensitive assay, which was a whole animal assay, using a gene that is not the gene that they're actually delivering. So they did 3 different types of characterization for biodistribution where the drug went. One is they ask the question, where is the luciferase expressed? But they use the least sensitive way to do that. The second one is that they put a little bit of radioactivity on the RNA, tritium, and they injected the radioactive RNA plus the lipids, and then they could see where the RNA went. And so when you look at that table that you're talking about, that's the right panel, is the tritiated RNA. And then they used an assay that actually detected the lipids present in the various tissues. And in that case, they did grind up the tissues. And they assayed where did the lipids, I'm sorry, are the fats we were talking about that coat the RNA. And these are novel chemicals. They've never been really used in humans much before. Okay, except in some cancer experiments historically. So what they found, which is what you're talking about, is that the radioactive RNA didn't stay put in the muscle where it was injected. It went throughout the body. Much of it stayed at the site of injection, but much of the radioactivity went elsewhere. And some of it went to spleen, liver, places you'd expect it to go that filter the blood, but part of it went and got stuck in the ovaries of the female animals. And furthermore, the lipid part, the fat part that coats these things, also went all over the animal. Some of it stayed where it was injected. A lot of it went elsewhere. And particularly striking was that 20% of it, approximately, ended up concentrated in the ovaries of the female animals, but it was not concentrated in the testes of the male animals. So something different about ovaries from testes, both reproductive organs in humans and in mammals, and it showed that there was this accumulation of novel lipids in the ovaries, and that has a lot of people worried. That doesn't mean that the spike protein is being made in the ovaries. We don't actually know whether it was or wasn't, because they didn't use the really sensitive assay for luciferase even, let alone look for spike protein, which they could have done. But they showed that these lipids were concentrated in some tissues that are of concern, particularly for reproductive biology. And so that's the nature of the data that you're talking about. And there's a caveat that we have to all be really sensitive to, I suggest. This is a data set that Pfizer submitted to Japan, presumably the United States, and EMA months and months ago, that they used to justify administering their vaccine to humans as an experimental product under emergency use authorization. So it's not yet a licensed product. They can't legally sell it. They can't put an advertising campaign. You know, you can't walk into your pharmacist and you see a little brochure there saying, you know, buy Pfizer. They're not allowed to do that, at least in the United States. This is the data that they submitted a while ago. I don't know exactly when. Now, they've submitted a new set of data to the US FDA just a few weeks ago. And hopefully they've addressed these things of reproductive toxicology, genotoxicity, biodistribution, using rigorous good laboratory practice methods. And I hope that they have. And I hope that those data show something different. The thing about that I caution is that those studies were not done rigorously. And so we can't criticize them, you know, given that they weren't done rigorously, we shouldn't overinterpret them. They may be right, they may be wrong. They weren't done rigorously. Now that is enough to be upset about, and I'm a little upset about it, and I don't think it's right, but it's what was done in the heat of battle. You know, during an outbreak, there are a lot of really hard decisions that have to be made on short notice in a crisis situation when people are filling up your hospitals and they're dying. And corners got cut. There's no question. But now things, I don't know about Portugal. I hope Portugal is in better shape. Certainly the United States is. And we have a little bit of breathing room and a little bit of time that we can do things right. And so that's hopefully what's happening. So I caution to not get, not assume that these data are the final story.
Okay.
Have I explained this to you? Is this good, what I said?
I understand. Yes, I think it's clear. Thank you. So another question that several doctors, I think in US, have expressed some concerns about the disease called antibody dependence,
Yes.
ADA,
Yeah.
possible long-term effects of the vaccine.
Okay.
That they are concerned that may appear in the autumn with the return of the other coronavirus. So what you can tell us about that?
Yes. So the term she's using is antibody-dependent enhancement.
Exactly.
Those three words are really important. And Brazil and Portugal have a rich, long history of interchange with each other, right? So
yes.
many Portuguese know about dengue virus in Africa and Brazil,
yes.
right? Yeah. So dengue virus is the classic example of antibody-dependent enhancement. And it is a very complicated topic. It's kind of complicated immunology. I'm going to try to make it easy by talking about dengue so that we can understand it. With dengue, it's not the first infection that kills you or makes you really sick. It's the second one. And particularly if you're infected with one strain of dengue and then later get infected with a different strain of dengue, then you can get really sick and you can get something called dengue hemorrhagic fever. Now that's a special case of antibody-dependent enhancement, but it's one of the best known. I'm going to really oversimplify this, so forgive me. To go into the full biology is a whole talk in and of itself. But if you get infected, in some cases, if you get infected by one virus or you get a vaccine against one virus, and then you get infected with a closely related virus that's slightly different, the antibodies that were generated, remember we talked about antibodies, they're these, it's like a fork. It's got prongs on one side. It's got a handle on the other side. The prongs are what bind to things like viruses. So here's a virus. Here's an antibody. It sticks. The scale is off. But I'm working with my hands. So an antibody can attach to a virus and can block the ability of that virus to infect. Okay. And that's one of the forms, one of the ways that antibodies can work. In the case of dengue, if you raise antibodies that are against the first virus that looks a certain way, and the next virus is slightly different, the antibodies that are made against the first virus can be good for neutralizing the virus and making so that it doesn't infect anymore, and your body clears the first virus. Then along comes the second virus, and it's slightly different, and the antibodies still bind, but not so good. And they're not able to really block infection. And in some cases, the handle part of the fork that's sticking off of the antibody, if you imagine this is the tines of the fork, sticking on the virus, but it's not really blocking the important parts of the virus, or it might be, and the handle of the fork is sticking out, sometimes that can help this complex of virus coated with antibodies infect cells that have the receptor for the handle of the fork, for the back end of the antibody, and suddenly the virus will be infecting cells that it wouldn't normally be able to infect. like macrophage, monocytes. These are cells that can migrate through your body and produce these really potent signaling molecules called cytokines, chemokines, and other things. And so you can end up with antibodies that enhance, make it work better, the infection, okay? So antibody-dependent enhancement of virus infection and replication. And that can make a situation where normally if you had the first virus and then you get infected by another virus or exposed to the same virus, these antibodies that worked against this one will work just as good on that one. But if you get a slightly different virus coming in, the antibodies that worked against the first virus may not work all that well on the second virus, but may work good enough to enhance its ability to infect cells that it shouldn't otherwise infect. And then you can get this explosive infection, and all kinds of things can happen. So that's a very simple version of an antibody-dependent enhancement, but I think it's good enough to help understand why some virologists, physicians, and immunologists may be concerned about what happens with the next virus infection. Now, why would they be worried about this with a coronavirus? Well, they probably know that all the prior efforts to develop a coronavirus vaccine, including for veterinary animals, there's a lot of agriculture in Portugal, I know, I'm glad of it, with cows and dogs and other animals that get infected by coronaviruses and have diseases like peritonitis that the coronaviruses cause. And so the companies that make veterinary vaccines would very much like to have vaccines that would prevent those things from happening to food animals and companion animals. And they found it extraordinarily difficult to make coronavirus vaccines. They've succeeded in a couple cases. But the problem is that they've had chronically, again and again, a problem with antibody-dependent enhancement of disease. So it's been a problem in veterinary vaccines. And when people tried to develop vaccines for SARS-1 or Middle Eastern Respiratory Syndrome virus, which is related, MERS, all of those prior attempts have failed. And one of the reasons they failed is because the vaccines that were developed caused antibody-dependent enhancement. Now, I personally didn't take a vaccine for a long time, when I developed the technology, right? And I'm a vaccinologist, but I didn't want to take one. Now, part of the reason was because I was infected with COVID in late February of 2020 in Boston. Okay, so I was among that first wave, and I had long COVID, and I didn't really need to have an immune response from a vaccine because I'd already got it by being infected by the virus. But in the end, I took it for two reasons. Well, really three. But the two main reasons was there was rumors that vaccines would help with long COVID. This is this long syndrome. And the other is because my wife and I wanted to travel to Portugal so badly, and we wouldn't be able to go if we didn't have vaccine. And that's the honest truth. We wanted to go back to Portugal and visit our friends there. So we decided to take the vaccine. And we had some side effects, but it didn't kill us. So there's that. If anybody says, well, why didn't you take the vaccine, or how come you took it because you know all these bad things? With all of these things, it's a risk benefit. The problem is that we really don't know the risks well enough. And there's some signs that there are some toxicity problems, and we don't know how frequently they happen and how severe they are. One of them is
my next question. What is the problem with the spike protein? What is the problem?
Okay, so with these gene vaccines, and you have to dial back to, they actually started making them. Tony Fauci signed the first contract with Moderna in late December of 2019. So as soon as they heard information from Wuhan, China, that there was this problem, they said, we better get going and we better try to make a vaccine as soon as possible. So if you've never been, well, now everybody has lived through it. But for me, I've been through many of these outbreaks. I was very involved in the development of the Ebola vaccine that became the Merck vaccine. And these big outbreaks drive people crazy. And I was very involved in the Zika outbreak and trying to help Brazil and help protect the United States. So these things really affect people. And when you're in the middle of it, at the front edge, it's really hard to think clearly. And it's a little bit frantic, as you might imagine. And sometimes decisions are made. Always, we never have enough information. And sometimes people make decisions quickly that they later regretted they didn't take more time to think it through. I think maybe that's the case with the spike. I speculate. Spike, in these little RNA viruses like coronaviruses, there's so much evolutionary pressure that these viruses evolve multiple functions that overlap in the same protein. And spike is an example of that. Spike protein does enable the virus to stick to cells by ACE2 receptor and to enter those cells and undergo a fusion event, but it does a lot of other things. And spike alone is active in modifying certain immune system cells, immune system proteins that make it so the cell can fight off viruses. It kind of turns those down. It signals through NF-kappa B and turns on TNF-alpha and other things. So spike is not just an antigen. It's biologically active. And among other things, free spike is known to cause damage to cells. So we call that cytotoxicity. And it's known, there's multiple, this isn't controversial, except in the press, but in the scientific literature, it's well documented that spike also causes this blood-brain barrier, which is the thing that separates our blood from our brain compartment and keeps bad stuff from going from your blood into your brain or things going from the brain out into the blood. So blood-brain barrier is like this filter kind of that exists in the cells and the underlying structures of those cells that course through the brain that bring oxygen and nutrients. So blood-brain barrier is really important. And spike causes this blood-brain barrier to kind of open up so that things can get through that shouldn't ought to get through. Okay, so spike does a bunch of things, and among the other things it appears to do, is be the bad actor that's causing the blood clotting. That's a problem with the novel coronavirus infection. So people that get COVID often have problems with their blood clotting and microvascular clots as well as bigger clots. And it looks like spike is kind of triggering some of that stuff. When they were engineering these genetic vaccines that cause your cells to start making proteins, they took the spike protein because it's very immunogenic, and they genetically modified it so that I mentioned that spike has an open conformation and a fusion conformation. Okay. And they genetically engineered it so it's locked into the open conformation, which, by the way, is the conformation that binds ACE2. And when spike interacts with ACE2, it causes vascular endothelium to do things. It also triggers activation of vascular smooth muscle. So then it clamps down the vessels. It's biologically active. and they engineered it so it stays in the open conformation so that antibodies can get to that, and you could build neutralizing antibodies. It makes sense from a vaccine standpoint, may not make such good sense from a general health standpoint. They didn't want the spike to go free in the blood, and so they put a little tag on the end of it that would make the spike protein stay put in the cell membrane. So going back to our egg, they engineered the spike so that they thought it would get to that edge of the white part of the egg where the little crunchy frying stuff is, the membrane, and it would stick there, okay, through genetic engineering. And so they put an membrane anchor sequence on it, and they engineered it for the open conformation. And they tested that a little bit, and they said, oh, it stays put. Hallelujah, we succeeded, we're geniuses. And that became the basis for the vaccine because it's very immunogenic. And they did some animal models, and it was very immunogenic, and it all looked good. And then and they went through and they started administering it to humans, and they had some adverse events, but the virus was so bad we didn't worry about them too much. Perhaps I can only speculate what went on in the mind of the regulators, but recently a paper got published, a nurses study by Harvard and Brigham. So these are very important academic institutions in the United States. And so they did a study with, I think, 14 nurses that took the Moderna vaccine, which is the largest dose, right? We need to understand the Moderna vaccine has much more RNA than the Pfizer vaccine does.
Okay.
So they took the Moderna vaccine, these nurses, and then the nurses agreed to have blood samples drawn over time. And very sensitive methods were used to detect whether or not spike protein was in their blood. These are probably the methods that Pfizer should have used instead of using luciferase. But so somebody at Harvard is smart, they figured it out. They used this really sensitive method, and they tracked whether or not there was free spike in the blood of these nurses. And in fact, there was. And in some of the nurses, it lasted for almost a month. It kind of peaked at about 2 weeks. And it wasn't, it's a teeny tiny amount of protein, that's the truth. When I did the calculation, just a sec. Interesting. Pardon me, a colleague texting me about vaccines and COVID. I don't know why he would send that to me. So they did this nurses study, and what they found was that there was free spike protein circulating in the blood of these nurses. And it came to about, if you assume that they had a blood volume of about 5 liters of blood and multiplied the concentration of the spike protein on average, it came to about 1/3 of a microgram. So that's a teeny tiny amount. You couldn't measure 1/3 of a microgram of sugar onto a spoon if you wanted to. It's a very small amount. However, for a very biologically active protein, that's not trivial. For me as a gene delivery specialist, I thought it was an enormous amount of protein, an enormous success, just from a functional side of the technology working. And one of the things about this study is that spike protein is the free stuff circulating in the blood, but there's also spike protein that's bound to ACE2. So forgive me, there's a little bit of biochemistry. There's free spike in equilibrium with ACE2 bound spike in equilibrium with the membrane anchored spike. And so a metaphor that really applies is the free spike is the tip of the iceberg. If you know that, right? Mm-hmm. You just see the tip, the big iceberg is underneath the water.
Yes.
So there's a lot more spike than just what you see in the free spike.
Okay.
So this is circulating the blood. And what does that mean? The honest answer is we don't know. But it's a little worrying because it wasn't supposed to happen. We went to all the geniuses that engineered this, went to all this work to make sure it stayed put, and it didn't stay put. it got cut. Proteases, little Pac-Man came along and cut it off of the surface, and then it floated free in the blood where it can do things that it isn't supposed to do. So that is of concern. Now, where it gets interesting is that this observation that, for instance, remember, I'm going to loop back. I talked about the adenovirus vectors like J&J and Sanofi, those vaccines that produce spike for a longer period of time. And we know that they have had problems with blood clotting, with coagulation. For instance, the Norwegians won't even let you use those in Norway. I hear from my friends in Taiwan that there's a real alarm because they're having problems with the J&J vaccine in their older people. And older people are having blood clotting problems. So we know that spike can cause the blood clotting problems.
And the placenta. Because I think Dr. Mike Hidden, I don't know, talk about that spike protein could attach to the placenta. That's a real problem or no?
It could attach to a lot of things. And so now we're talking about adverse events. And another adverse event that many women report, I hope this is okay to talk about this in Portugal, but women report problems with their periods after vaccination. Sometimes the flow is very heavy. Sometimes the timing gets changed. So that's also related to reproduction and the female reproductive system. We don't know. The problem is that we don't have enough information that is good enough to be able to say for sure that certain things that normally happen at a certain frequency, even death, are related to vaccination or not related to vaccination. But there's enough reports that many people are a little worried that there might be a relationship. And I'm trying to be very careful. I want to be able to get into Portugal and not be stopped at the border.
No.
So I don't want to say things that will get people upset.
We need tourists.
But there are concerns, and it used to be that most governments were telling the people that there's nothing to worry about, that there's no toxicity with these vaccines. But then some people, at first the FDA, then the CDC, then in Israel, found that there was some problems with hearts, particularly in young people, young men. And they had thought that there wasn't any problem like that. And of course, they saw that there was problems with blood coagulation with the adenoviral vectored vaccines. And there was a period of time where those were stopped in the United States. And in some European countries, they're still not being used for that reason. And that's a spike problem. And now there's talk that one of the things that the vaccines may be doing is turning on latent viruses so that sometimes people will get shingles after and I can tell you I've had shingles, it's extremely painful. So activation of latent viruses may be another one. These concerns about menstrual cycle and menses and things they may be real, they may not be real. Hard to say, got to do the statistics. Another one that might be real is thrombocytopenia. Very low levels of blood platelets, which the FDA knows is a potential complication anytime you administer polynucleotides. Polynucleotides is the science word for fragments of DNA and RNA.
And to one person who already have thrombocytopenia, that could be dangerous. Or no?
Yeah, it could be if you already have that. And So there are these things that we were all told none of these things happen. And then over time, as we have more data, then we find out that in fact, some of these problems do happen. And the problem is now, who do we believe and trust? How serious are these things? How often do they happen? And what can we do about it? And it has many people worried. I want to say something to your audience. I'm not saying don't take the vaccine. Please don't take that conclusion. These vaccines have saved a lot of lives. What I want to say just for a moment, something about bioethics, because these remain experimental products. Here in the West, I can't speak for China or Myanmar or other places that have authoritarian leadership. But here in the West, in Portugal, in the EU, in the United States, we've all agreed on certain things that have to do with human rights and medical research. It goes back to bad things that happened during World War II and the Nuremberg Code, and has been further developed in a body of scholarly research that we all agree to, which guides what is ethical to do during clinical research and what is not. And there are three basic fundamental things having to do with research ethics. And why do I say research ethics? Because none of these are licensed products that we're talking about. They all remain experimental products. So they're all, the administration of these is technically medical research still. We're still gathering data, just like you and I have been talking about. And so there are some basic rules if you're going to administer an experimental product to a human. The first thing is there has to be full and open disclosure of adverse event risks. They owe it to us by the rules that we all agree on to fully disclose anything that might be worrisome. Now, some governments are afraid of doing this because they think we're not, frankly, it's kind of like treating us like we're children. They think that we can't think through and realize that maybe getting a headache or getting dizzy, the risk of those things happening, which are rare, or even thrombocytopenia, very rare. But COVID, if you get COVID, it will change your life. My body will never be the same. And it can kill you. And it can kill you at about 1 to 2% of people that develop it, and higher if you're older. So I think we're all grown-ups, or you and I are, and we can make our own decisions about risk. But whether or not those are the rules, is we have to be given the information about risks so that we can make an intelligent decision about whether or not we want to accept those risks. Public health authorities owe that to us. It's not just theoretical, it's the rules for experimental products. Rule number 2, those risks have to be described in simple language that we can all understand, that the peasants working the farms can understand. The gypsies can understand. It has to be said in a way that all adults can understand it. We can't hide behind fancy scientific words. So all the risks have to be disclosed. They have to be disclosed in a way that adults can understand them. And then for adults, they have the right to accept or not accept the experimental product. It's bedrock. They have the right to not do that and not be pushed or coerced to do it. So this means that we're generally free people. We make decisions about our lives, and my position is that the state, the government, owes it to us to convince us that it's the right thing to do with the information that they have. They have amazing tools to convince us to do things. They're not hampered in doing that. They don't have to force us to do something. They should teach us. So that's my position. And then for the children, babies, children, young adults, adolescents, up to the age of consent, we call it that because that's what it is. We all agree that they don't have the ability, the mental faculties to decide these things for themselves about whether to take an experimental drug or not. Only their parents or their guardians do. That's just the way things are. And so children, adolescents, and babies cannot give informed consent. Only their parents can or their guardians. And so it's really not okay to, as some countries have done, I hope not Portugal, but try to get children to take a vaccine that's experimental without getting their parents' approval. That's just wrong. And I think we all know that. And in fact, today, the World Health Organization has announced that they're not recommending that children be vaccinated for COVID.
I saw.
So yay.
Yay.
So I think many of us have had real concerns about children and adolescents because they don't generally get severe disease and they do have risks from the vaccine. And so this fancy sciencey term risk-benefit ratio just kind of doesn't make sense for them. And now I'm glad to see the World Health Organization agrees with that. So I wanted to say this about bioethics, and this is the reason, the thing that's so problematic about my own government not being completely open and transparent and honest about what the adverse events, the potential problems are, how severe they are, and how frequent they are. I'm not saying that these are not safe vaccines. What I'm saying is that the government owes us a full disclosure of how safe they are. All vaccines have problems. All drugs have problems. I'm arguing that not that you shouldn't take it or that it's not safe, but rather I'm saying we deserve, and the government is obligated to share with us the information that will allow us to make our own decision about whether or not we want to accept this vaccine, that vaccine, or no vaccine.
And open discussion, right? Not censorship.
Yep. Amen.
So one last
yeah.
question. You shared
Yes, ma'am.
the article in your Twitter about how much jabs a person needs.
Yes.
I want your opinion. One person that already have the disease, they need to vaccinate it. They just need one jab. What do you think about that?
I agree.
Yes.
And a colleague of mine that I have helped mentor from time to time over years, we first met during the Ebola outbreak, who is a senior officer in the U.S. Army and was very involved in helping guide the development of the Moderna vaccine, asked for a Zoom meeting with me last week to discuss these things. And he wanted to learn what I thought, just as you were asking, about a single jab if you've already been infected. And I told him the same thing I'm telling you. I think it's a good idea. They should really look at it. But it needs to be data-based. We need to stop making decisions on the fly. They need to be based in data. But I think the data are there to support the idea of only taking a single dose after you've been infected. And I think that the data may be there
But
that we
to
don't
all
have.
people, or just I don't know, the older, that the immune system is weaker?
Certainly, there's a stronger case for the older adult who is entering immunosenescence, like me. That is fairly clear. What the data also show is that the adverse events are more and more severe with the second dose. And so in the person that's already infected, they already have some immune response. Having only one dose, I think, makes sense, but it's not my decision. It's the decision of your health authorities in Portugal and in the EMA and in the United States and at the WHO. But I think that they should strongly consider a single dose if you've already been infected. And I think that there are data suggesting that for all of us, for some of these vaccines, one dose may be enough. And the CDC was supposed to meet last Friday to talk about this, but there was a new federal holiday, we call it Juneteenth, that just started this last Friday, and so they canceled the meeting. So hopefully that meeting will happen this upcoming week, and then hopefully this time the CDC will have done its homework and do more rigorous assessment of risk benefit for these questions: one dose versus two dose, adolescents, children, babies, pregnant women, very old, healthy, normal adults, and do the calculations for each of those special groups to be able to say, yeah, the vaccine makes sense here, maybe it doesn't make sense there. So that's what they're supposed to be doing. And I think the world is waiting to see what the advisory committee on immunization practices will say, hopefully this week later, but maybe not, I will see. So have I answered that question?
Yes. So thank you so much for joining me. There was a very interesting interview, and I learned a lot, and I appreciate your kindness. Okay.
And I hope that we can meet sometime soon in Golga for the horse fair.
Golga, yes.
Okay.
Yes.
Okay.
So thank you and bye. So.
Okay. Bye-bye. Thank you for your time.